Physics at, at, at Weill Cornell Medicine, as well as director of the World Quant Institute for Quantitative Prediction. This lab has a wide array of interests in genomics, including cancer biology, epigenetics, and microbiome research. Their prolific contributions to science have been recognized by numerous awards, including the NASA Group Achievement Award, the Pershing Square Zone Cancer Research Alliance Award, and the Herschel Weil Collier Career Scientist Award, among others. Chris leads the International Metasub Consortium, which is doing some amazing work in environmental metagenomics, as well as microbial surveillance. And it's a great example of international collaborative science, bringing together scientists from around the world in a extremely collegial and collaborative fashion. His talk today will take us on a whirlwind tour of SARS-CoV-2 microbial surveillance and onwards into space biology, highlighting all the key strengths of genomics and some of the amazing work that's happening in his lab. So thank you, Chris, for sharing this with us, and over to you.
Thank you so much, Dr. Nigarajan. It's a pleasure to be here. I wish I could be there in person, but hopefully at some point I'll be there physically once the pandemic starts to come to an end. And so can you see and hear me okay on slides and audio? Sounds good?
Yes, sounds good.
Wonderful, great. So I'll take you on a bit of an adventure of everything that's happened, looking at viruses over the past really 16 months and everything that we've learned, and also what's coming in the future, both obviously for things we've discovered across dozens of cities here on Earth, and looking ahead a little bit to some things that someday we might find actually on Mars. Okay, so I'll start with the fact that I think it's something that's certainly probably not lost on anyone here, is that viruses are everywhere and really an engaged and important feature of our ecosystems, from plant microbe interactions to infectious disease to really mediating a lot of the facets of our biosphere. And so what's interesting, of course, as I think we've all learned, and maybe you're tired of hearing COVID talks, but this one will be a little bit different because it'll link to a lot more, not just about COVID, but how do we think about using SARS-CoV-2 as a model by which we could learn about really any virus. So in particular, when the pandemic broke out, I drew out kind of a version of this on our whiteboard in our lab, is that we started sequencing patients as they came in through the hospital. And we thought, we really need a battery of methods to really understand this new virus and what's the host response and even microbiome changes. And so the ones I'm going to walk you through today are ones that, I've worked with companies, I've worked with nonprofits, worked with academic groups. I'll just disclose any conflict of interest whenever one is there. But basically, we used every possible method we could to understand what happens when you're encountering a new virus, everything from the virus to its genetic variation to the host response. And so I'll start with these two in red. Is one thing we do a lot of is RNA sequencing, and have been doing it for decades at this point, is we wanted to deploy some of these newer methods and also compare it to RT-PCR, because they're fast, but also very informative. And so this kind of gives you some details of how long things take. But in particular, what happened is really for the very first patients that came through the door, it was 857 samples. So we did some patients twice and wanted to take them that had been suspected of SARS-CoV-2, grab those patients and say, could we look by RT-PCR, shotgun RNA sequencing, and LAMP, which I'll come back to in a little bit, and compare these methods and see what the differences between these patients and what we could learn. So we found out that actually 216 of them were confirmed positive by RT-PCR. Now, remember, these were all suspected COVID patients, so they had some sickness, and I'll come back to that later, but also some environmental sampling, which I'll show you, and also some controls. And this work was recently published in Nature Comms. You can see the reference down at the bottom. So the first thing I want to do is take the clinical samples, these nasopharyngeal swabs. You take that swab, we shatter all the cells and extract the RNA, and then sequence everything that's there, and then map it to its read fate. So what is the fate of each sequence of RNA that we looked at? Where did it end up? So in particular, we looked in the samples that were RT-PCR negative or positive, and some of the negatives actually did show up a fair amount of the virus. You can see here in orange, whereas the rest of it was human RNA in blue, bacterial RNA in green, and if we're curious to see these positives, you can of course see some high, medium, and low amounts of the virus of SARS-CoV-2 as compared to some controls here on the left. But you could see it was mostly human bacterial and viral RNA, in these swabs. But one of the first things we want to look at is there evidence of other viruses being present? So we took all the fragments of RNA and said, okay, we can confirm SARS-CoV-2 is present. What else is there? We could see some had a lot of SARS-CoV-2, but what else was there? we can see that co-infection was actually quite rare. You can see this is the low, medium, and high amounts of the virus by sequencing and RT-PCR. Over here is none, meaning there was no real detectable amounts of SARS-CoV-2, but we could see evidence of other viruses, other coronaviruses, rhinovirus, RSV. We could see these other viruses present. And it really showed that co-infection was only about 4 or 5% of patients. Otherwise, if you had one infection, that was generally what was causing your ailment. But we did see some co-infection with flu and SARS-CoV-2. And to confirm these, we used another assay called BioFire to see how well could we do with RNA sequencing if this was a diagnostic assay, and actually did quite well for the respiratory pathogens. You can see our sensitivity was only about 80%, but our specificity was very high and overall really high accuracy to take shotgun RNA-seq data and then be able to validate this and use this essentially as a diagnostic, which we're working on now and rolling this out. So that was at the viral side. Then we wanted to look at, well, what about the microbiome? We have basically a swab of someone's nose. Could we look at the microbes that are present as well? So indeed, we could see that for the normal flora, these are basically, every one of these lines is a normal commensal bacteria in either the oral passageway or the airways or shared microbiome between the two. And these are basically bacteria you would expect to see, but we saw in the high viral titer patients a microbiome disruption that led to the alteration of these particular species significantly, statistically in red, that were really associated with the infection, which was also interesting to find out. So then we looked at virus, we looked at bacteria, then we wanted to see, well, what about the host? What about the human RNA? So remember, if you go back to this slide, you could see all this RNA is used human in blue. Well, what happened to the human genes? So we looked for differentially expressed genes, or DEGs, that were statistically significant, also at least 1.5-fold difference for things that were upregulated here in orange or downregulated here in purple. And you can see the statistical significance on the y-axis and the fold change here on the x-axis. So basically showing that there's all these genes upregulated in response to infection, which include some genes you might recognize, like interferon-stimulated genes or some IFIT1 or IFIT6. These are genes that are known to be associated with a viral infection and response. And so when we do a pathway enrichment to see what are the types of genes that are activated upon infection, indeed, these are what we see. We see beta signaling essentially for interferon, type 1 interferon, viral response. So these are hallmark signatures of being infected and what your cells do to defend and your immune system does to defend it. But what was surprising, these new networks included things like olfaction or hemoglobin or oxygen binding, very unusual pathways in response to infection. And so what's interesting about this, these are the genes being down-regulated, so turned off in response to infection. And this is what's leading to what we think some of the anosmia in these patients, or the loss of sense of smell or taste, we can pick up effectively, you can see right here. And so one of the things we want to see is, well, can we make sort of plots that would show these genes that are going up and down and make an interface so anyone could pop these out? So actually, Dr. Maiden in our lab put this together. This is something that came out as a resource for the paper. I'm also going to just show it here. This is COVID genes.wild.cornell.edu. So you can actually see what's on my browser. You can see what's normally sitting here. And basically, if you go to this website, it should pop up here any second. Here we go. So this is some details about the study, as well as some autopsy data that we've looked at. And this lets you basically take, for any given gene, you can fill in a given gene and pick any gene of interest. It'll make a violin plot of what we saw for these data. For example, high, medium, or low amounts of the virus, or what do we see for nasopharyngeal swabs across other cohorts, and then also what do we find for patient autopsies. We can see here in the different parts of these postmortem tissues. And you can also make heat maps if you want to add different genes to your heat map and adjust this. Here is just a simple R-Shiny server interface, and you can also look at pathways. So we built this as a way so that people could browse and characterize the data on this website as a general resource for the field. You can see here, again, published already. And so let's see, can you still see? Okay, great. I'm going back in. Okay, you can still see my screen, I think, yes? Great, okay. So if anyone wants to browse the data, please do so. It is a resource for the field. So the next thing we wanted to look at was also some spatial transcriptomics. And in this regard, we worked with NanoString to actually use the genomics platform, which was to look at 39 autopsied patients in these regions of interest, working with Rob Schwartz and Alan Borsick, 2 doctors. We performed the autopsies, were wonderful collaborators, still are. And we see you can basically take UV photocleavable oligos, look on a slide, and actually once you find what you want to look, you cleave them off with UV light, you aspirate these oligonucleotides, put them into a well, and then you can sequence them or count them and actually generate this high dimensional profiling of different tissues taken from patients, either from cancer patients, or in this case, we do it from COVID patients, unfortunately from COVID autopsies. And what we could observe with these data, with this genomics platform, was we could see high TMPRSS2 in both the large airway and the alveoli. You can see here, ACE2 receptor is kind of this ring of the canyon of the alveoli, and DAPI, the DNA stain here in blue, and you can see TMPRSS in red, and then again, the virus occasionally showing up, but generally was at low levels. Actually, by the time the patients succumbed from the virus, we found that often there was very little virus left. But what was interesting was not that there was a lot of virus left, but again, these patients died for some reason. What we observed was that actually there's a massive infiltration of immune cell types that are not normally present in lungs. And so in particular, these are some of these COVID patients. Each vertical stack of lines is a region of interest where we sequenced and profiled the cells. And you can see here a big burst of macrophages or monocytes or neutrophils or plasma cells in particular are not normally found in lungs, but they are suddenly present at high numbers and led to a lot of infiltration and disruption in these lungs, and unfortunately likely contributed to their demise during the course of the infection. And so this shows that really is a distinct disease in terms of what it does to the lungs, how they respond, and what's happened in these patients. This also, again, was just published a few months ago. Okay, so that is looking more at the profiling of the host, but there was still, in the early days of the pandemic, really not that many good ways to do a capture or sequencing of the virus. And so we worked on with Twist and with Biosha, and full disclosure, I'm a co-founder of Biosha. We worked to get emergency use authorization of a hybridization capture in next-gen sequencing assay. So you can see here, the way it works is you can collect a specimen, you basically put it into a mini prep kit, can convert to cDNA, prep it with a library enrichment kit, get the probe panel, and you can sequence it. We often do it on a NextSeq 550. And then you can do an analytical pipeline here to see essentially what are we picking up. And so what we could see in this sort of 5-module pipeline, which takes about a day and a half, but you get really high sensitivity to different kinds of viruses. So in particular, we can find known viruses down here. This is the percentage of known samples of SARS-CoV-2 that have a particular variant, like some of these variants listed here. And we got it down to about 96% positive percent agreement and about 100% negative percent agreement. And this was enough for the FDA to get a validation of this. And so what's exciting about this is that it worked well for catching the virus. Also, you can see the preprint is online here. But it also let us find that we could get pretty good coverage across the majority of samples. Once you get to CT values below 30, you're fine. But if it goes too much higher, we would find in some cases that you'd lose some coverage, but this is not too unexpected. We saw the same thing with shotgun RNA-seq as we do here with sort of the hybrid capture methods. And so the thing I was really excited about those, we do get complete genome coverage, for these variants. Even a large deletion like in the 117 variant, we could pick that up. So known variants could be picked up. But I think even more excitingly is that some of the novel genetic variants we could pick up with this assay, because the capture probes are kind of like Velcro. They're so long, they're 120 nucleotides. It gives us a large bait by which we can actually capture novel variations of SARS-CoV-2. And so this is, again, the preprint is there on the bottom, and some work that we've done there with Biosha and with Twist. And finally, I'll just point out as a placement and frequency report. So if you get this report, we also built up an interface so you can see where is the sample, how often is it seen, for example, in Singapore or in other places around the world. Okay, so we're not done with our diagnostic tour yet. One of the last places we want to stop is here at LAMP. And so if you've not heard of LAMP, it is a loop-mediated isothermal amplification. This is an assay that's been commercialized by New England Biolabs. And so we began using this back last February because no one could get PCR primers or reagents. So we thought, well, what about LAMP? And if you haven't heard of LAMP, it is a cousin of PCR insofar as you use primers for a target DNA or cDNA, but there's 6 primers in one set versus 2. And what you can do though is put this all in one tube. You can have basically a strand displacing polymerase with a warm start reverse transcriptase, quickly generate cDNA and then amplify a target, all within about 30 to 40 minutes. So when we first got some of these samples, we're using synthetic RNAs from Twist to actually see, can we get 0, 1, 10, 100, 1,000, log 10 increases in the dilution of this RNA, and say, okay, the reaction starts all pink. There's phenol red that's in this reaction. And basically every time you incorporate a base, if you go back to this image here, you essentially release one proton. So essentially the longer the reaction goes, the lower the pH becomes. So after about 15 minutes, the vials, the tubes with the greatest amount of virus got yellow. And within about 30 minutes, those that had even 100 or sometimes 10 copies, we could pick up the virus quite well. We were very excited. It seemed like it was working. We then tried this across replicates and across clinical positives. We found we could pick it up there as well. So we quickly started releasing the protocol. I was emailing this to everyone I could, started working with color genomics. And also we got the first green light for a LAMP-based COVID test, in this case, detecting positives, now widely used across San Francisco. We also got this set up so it is a full kit with New England Biolabs, and I helped them as an advisor for this product that was coming out to market. So now anybody can use this kit for SARS-CoV-2, but what's interesting is this has become really a global group. So we released the protocols and the methods and the tools with everyone. It's now a global group called the Global LAMP R&D Consortium of everybody working together to sort of, basically come up with new protocols and have the best practices and methods for LAMP for not just COVID, but really for any target. And again, there were many places that didn't have enough testing. So last summer, I went to my hometown in Racine, Wisconsin. My brother here is in the middle. He is the mayor of the town. Again, they had no testing at their health clinic. So we went and we set everything up there, and we taught the firefighters and the staff there how to use a test on saliva and run LAMP there on site. And so in particular, you can read about this here in this article. It said we got things set up with this Wisconsin city. The lab that we did this in, it looked like this. It looked a little bit like either a bomb shelter or a meth lab or, well, kind of an unusual place to set up a lab, but they had no testing. And so it actually worked quite well to get things set up there. And we got testing set up for the firefighters, for the community, got things set up really well. And then also got this set up at New York Presbyterian at our hospital and made it so we could start doing rapid screening there for everyone as they'd walk in, basically. So the thing that's really interesting is it really begs the questions. That's the end of the sort of first half of the talk, is this kind of tour of different diagnostic methods that we got through the FDA and got up into the communities and into my hometown. But it begs the question of, what if we had been looking all along? What if we had been continually surveilling for new viruses or learning about our environment in a better and faster way? And this has led to some work to looking into sewage, doing sequencing of sewage in Florida, Wisconsin, New York, and also with the Metasub Consortium, which you heard about at the beginning, and I'll tell you more about this shortly, is to begin to link sewage happening here with what's happening in broader efforts. So this is myself, but also my other co-PIs, Stefan, Helena, and George, all working on this. We wanted to build up something that would be a real molecular dashboard for the states and the cities who are tracking essentially what's there for hospitalization, deaths, cases, and the clinical features. And so this dashboard is publicly available. You can go take a look at it. We also want to link these data to what's happening on campus to get essentially academic and other clinical and metadata put together across the campus. So what we do with all the samples now is they come in, we basically pull them out, do basically this, turbidity testing, a coliform culture, do a purification. We do LAMP, which I just showed you, PCR, and eventually we do RNA sequencing and also strain identification. And what we've seen so far is pretty extraordinary. Actually, you can see here that across really the past year, we just got some more data on this last night, but the trend is the same, is that the positive caseload here in orange really well mirrors what we see for the abundance of copies per liter of the virus in the wastewater. So the wastewater itself is a pretty good metric and mirror of what's happening in the population, and in some days even preceded what happened in the population by a few days. And so we could actually use this as a signature to track what is the presence of this virus. And this led to some discussions with the Metasub Consortium, which you heard about before, is a global consortium that we meet every month and do sampling every year, which I'll show you in a second. But we had already been doing work collaboratively for several years. And we thought, well, what if on a call in March, we thought, let's all start to work together. So we set up a virus tracking kit to look at really RNA viruses around the world as they emerge, and not just SARS-CoV-2, but we want to look for really any virus, right? So this is kind of the snapshot of the consortium. And we could actually see quite clearly that if you look across all the samples collected, the number of reads we're picking up in each city was really dependent on where they were in the pandemic. Some of these samples are from hospitals and they're anonymized right now, but we picked up not just SARS-CoV-2, but we could also pick up influenza, metapneumovirus. We could pick up other viruses in the RNA. And if you look across the SARS-CoV-2 genomes here on the right, sometimes it was very fragmented. This is a coverage plot that shows what's the density of sequence reads we got across the genome. Again, sometimes very fragmented, other times very consistent. And so this really, this was looking at the RNA level, but we were inspired to kind of begin Metasub because of some work that we had done way back in 2013. So let me take a pause on this idea and jump backwards in time to how this all kind of began. is, we have this network of people swabbing around the world, but it started a little bit of pure curiosity. So back in 2013, I had a bunch of summer students at Cornell, and I said, what if we built a genetic map across all 5 boroughs of New York City and swab and sequence and annotate and timestamp and geotag everything we find and build a genetic map of the city for the first time ever? And so what if we pulled these all out, got 10, and we did all this and sequenced about 10 billion fragments, did alignments with BLAST and Metaflan, and then found that actually on average, about half of the DNA that we tried to map, it didn't match anything that had ever been seen before. It was completely novel. Whereas in other cases, bacteria were present or other organisms you can see, but it really meant that half of the world under our fingertips is unknown, has not been seen before, which really was, surprising, but then became actually very pleasantly haunting. And so I kept thinking about this of who is there and what are they making? Like, who are these organisms? And we published this in 2015. And we could see that we just wanted to know, are they making new molecules? Are there new species there? What are these organisms that are present? And so I kept getting emails and calls from various people around the world that said, I wonder what our city looks like. Does it look like New York City? And I said, well, I don't know, but let's start to work together. And so we actually, with World Quant and the Gates Foundation, began this project in 2016 in earnest. Was to begin, our original goal was to basically do, tracking across the year and just across several, about 20 different cities. It's expanded dramatically. And so instead of swabbing the world with one big swab, essentially we built out now to be a global consortium that builds metagenomic maps, antimicrobial resistance maps, and also we mine these data for new biosynthetic gene clusters, or BGCs, which effectively could become new drugs. And I'll come back to that in a second. So what we do is, every year as the Earth begins to pass the international dateline, the world starts to rotate. And you can see here that all the cities together on Global City Sampling Day, especially including Singapore, which does a fantastic job. You can see here that all the groups work together and also collaborate as the Earth rotates and people wake up and begin swabbing their part of the world. We have this occur again on an annual basis. And so since 2015, we started in the pilot data, and we've been doing since 2016. We just completed it a few weeks ago for this year's Global City Sampling Day. And you can see people get very, creative. Some people, for example, Dr. Nagarajan's team, you can see here, really creative in the lab, very good on Twitter. And you can see here other people even swab. This is Alina. She once got detained by the police, but then before she left, she said, I'd really like to swab this area before I go. And she did that. And so what is it? What are we doing? What's in the data set? So this has become a global effort of both DNA and RNA sequencing. And we just published some of this work a couple months ago in Cell of saying, what did we find? Sort of this first big snapshot across the world. And what we've done for this project is basically done a reference-based metagenome analysis, as well as de novo assembly across all these species, and then characterize them around the world, again, relative to all known genetic sequences. And so in the paper that just got published, we actually found that we were picking up almost 4,500 known taxonomic groups by species. And actually, this is across the number of samples. So the first batch of almost 5,000 samples across 60 cities, it showed that we're picking up thousands of species. But if you ask the question, how many species are in each city, or what's the prevalence of a species, you can see over here on the right that there were actually about 61 species that were in 95% of all cities. There were actually 31 species that were in 97% of all cities and all samples taken from all cities. And there were a lot more that were rare, these peripheral, we called them, that they would show up relatively rare. And this is a prevalence of different species. And if you look at what these core species, these ones that burst over here, of ones that consistently appeared, what they are is, some that make sense. They're commensals. Some of them form film, some are gram-positive or gram-negative, but a lot of them are not really commensals. They're things that are just other versions of species that have adapted to metal and stone and plastics and this sort of unique urban environment, which we called, the urban core microbiome. And what we can see here is that, there's really, it's not only can we find these different species present, but we can also track their antibiotic resistance markers. And so what we can do is for each city, build an antimicrobial resistance profile, or AMR profile, and see for each city what's the number that are detected, what's their density, you can see, excluding zeros, and what's the type. So in particular, what we could observe is that tetracyclines, you can see in some cities are very abundant, but here on average globally are near the middle. whereas we could see bactericidin or fusidic acid were much more abundant in general around the world. And so every city you can see here has this pretty distinct profile of what they look like for their AMR markers and their AMR distribution. But what's interesting is you might think, well, okay, that's interesting. Is that a lot? Is that high? Is that low? What does that mean? Does it relate to the population in some way? So in fact, it does. What we can see is that the over-the-counter antibiotic use can be derived from the cities themselves. At least from the data we've taken from the air microbiome, we can see here that essentially there's a significant association on both chromosome-derived and plasmid-derived AMRs, is that the greater abundance of use by the population mirrors what you see for the abundance of that molecule in the subway systems and in the air of these systems. So that's one interesting connection. The other thing that's interesting about this, though, is like you think, well, where else could they be coming from? What else might this indicate? And so we could actually see, would you find it, for example, like in a hospital? And so there's actually some great work by Dr. Nagarajan, as you can see here. Actually, I still have the preprint, but this is now published in Nature Medicine, is looking in the hospital across 179 sites. Do we see some of these AMR markers in hospitals, and what are they? And in fact, we could see that multi-drug resistant strains appear, and in some cases are even enriched in hospital environments. Again, because we think, as demonstrated by this work, that there is a selection pressure that is actually creating a way that, again, bed rails and bedside lockers, enabling some organisms to probably survive even better in these harsher environments. And so we've been looking globally now. We have a map that's called Resistance Open Map, where you can look across anywhere in the world and see, what's the abundance of these AMR markers. Now, again, to put this in context, you think, well, is this a lot or is this a little? We find, at least for what's in the subways, that it is 10 or even sometimes 100 times less than what you'd see in the gut microbiome samples, or what you might find in the soil. Is that if you compare to some other published work, it's not as if these are a hotbed of AMR markers, but they are present, but you can find more of them even in your gut or in the soil. So we don't think they're necessarily dangerous, but they are distinct to each city and they are different for sure. And so what these data have led to is a really extraordinary increase in the size of the tree of life, about threefold increase, and also detection and discovery of about 11,000 new viruses and almost 1,000 new bacteria. And so the other thing we found is also about over 800,000 new CRISPR arrays, which are the bacterial defense systems against some of these viruses. So we've actually been able to use this molecular echo of what the bacteria are fighting as a way to validate these new phages and new viruses that we picked up. And so this shows you these different types of CRISPR arrays that are present in our data. You can see here quite clearly. And what's also extraordinary about this is there's other things we can pick up from the de novo assemblies. In particular, you can look for new peptides or new biosynthetic gene clusters. And indeed, we found about 4.3 million novel peptides that are at least 20% divergent from anything that's ever been seen before. And so this is, the novel biology and really an extraordinarily, rich catalog of new species. So you often think of a rainforest as a place to go find new species, but you can see Tokyo, Rio, Oslo, you can see all these different cities have a lot of new peptides, particularly in Tokyo so far from what we've seen. So one of the things we also want to do with these data is say, well, if there's so many differences between cities, could they be predicted? Could they predict where you're coming from? And the answer here is yes, actually. So what we did is we did a training exercise to see how well we could do in terms of predicting, give me your shoe or give me a sample, could I tell where you're coming from? And we get to almost 90% accuracy with the species information, but it's better than the AMR. So this got cut off over here, but you can see the AMR genes were not as accurate. And it's because they are much more mobile. They move much more quickly around the world. Really are much more, unusual, and mobile elements. So they're less predictive in that regard. But it shows that there is this ability to predict, especially as you get more samples, where you're coming from the world. It's not going to solve a crime quite yet, but it does show it does have this predictive capacity, and that we can find new species, really all over the world. And so what's exciting about this, we're using these exact same methods, not just to find new species on Earth, but we're also finding some of them in space. And we've just actually found and cataloged a range of new bacteria that can survive in the space station, might be useful for trips to Mars. And we've also been working with the Jet Propulsion Laboratory, which is the facility that builds spacecraft at NASA. You can see here, this is the room where the rovers and the spacecraft get built. And as you go around, this, for example, is the Perseverance rover, which just in February got to Mars. You can see here the engineers are going around checking the instruments. And, they're people. They might get excited because they're sending a rover to Mars. So they might take a selfie with their robot that they're going to send over, which is understandable. But in the terms of microbial contamination, microbial effusion, you can see here, these are not hermetically sealed suits. As part of the skin microbiome, other air microbiome elements could go there. And so what's interesting is what eventually the Perseverance rover will do is it's going to be packaging up what are called Mars sample return missions. It's going to basically try and use this device to go around the landscape of Mars and collect some samples and then bring them back to Earth. So basically its job is to poke around Mars. This is a beautiful image of one of the craters in Mars. Look around maybe near some of the mountains on Mars. This again is a picture from Mars that looks very much like it's on Earth. Look towards the ground, find a spot where it thinks it can get a good dig into the Martian regolith, which looks just like this, and then basically capture these packages of soil and put them in containers, and then get them ready, look longingly back at Earth. This is kind of a new view of what Earth looks like if you're on the surface of Mars. You can see this about when it's 180 million kilometers away. That's what Earth looks like. But what it'll do is it'll sit there and wait, and then approximately in the year 2032, the Mars sample return mission is supposed to work with the European Space Agency and then bring these samples all the way back to Earth. And so what's really cool about this is we're going to have a chance, probably in about 10 years, to really examine biochemically, geologically, look at what's in these rocks, even genetically, try and understand what's in these rocks, characterize them, and maybe we'll find life on Mars. Maybe we'll see DNA or RNA molecules with RNA or DNA sequencing and see what's there. But when we do this, we do have this kind of nagging question, is that what if we send something all the way to Mars and then bring it back and sequence it, and all we do is find microbes from these people that were there building the robot? Is that possible? It's unlikely because the JPL is very careful. They build it layer by layer in extremely sterile environments and clean environments, but it's never 100% clean. So what we've developed is multiple ways to deal with this problem. One is, of course, we tell everyone to be careful. The second is we actually build a genetic profile of this clean room, which we call, this planetary genomic profile. And we have published some of this work recently. It's a metagenomics framework for organisms that can possibly survive the trip to Mars and back, as well as anything that might have been found in the clean room at JPL. And so what this means is with this method, we can find these novel species, just like we do with the metasub data. We do de novo assembly and look for new species and annotate them and look for their characteristics of what defines them, what makes it so that these organisms could be understood and placed in the context of where they could possibly survive. But that's at the annotation side. The other thing that we've done and that we've built with the metasub framework is work with Andre Kallas and Gunnar Reich within the metasub group, is we built a global camera index with this planetary genomic filter of where are all the different sequences that have ever been observed on Earth and what do we know about them. So I'll show you a quick preview of this. You can see here, again, working with the group, with Gunnar's group and Andre, as you can see here, all these samples, this is the metasub data set, but we also looked every other known sort of sample that's ever been collected, characterized, put together. And what you can do now is you can search basically for any sequence you ever want to know about, search across a different database. You can start with metasub or other databases. And discern, has this sequence been seen before? And if so, where has it been seen before? And what does it essentially look like? You can download the CSV, you can play with the data, you can see here. And then you can also do alignments. You can say, well, where has this been seen before? And then what does it match to query database? Visualize the De Bruin graphs if you'd like to, and you can kind of play with this graph a bit here of what's the connectivity between these different genetic elements. And so what this means is if you go back to what we see here for this camera index, just to go back, everyone can see my screen again, right? I hope. Yes, I'll take. But what this means is we have really a full genetic map of everything that's present around the world, and anything that ever comes back from a different world, we also can see. Just to confirm, I think you can see my screen, right? Dr. Nigaran, right?
Yes. Yeah.
Okay, great, just making sure. Okay, good. Thanks for coming back. So what this means is, this gives us a bit of a guilt by association. So even if we don't know what a sequence does, we can likely be able to tell, well, who does it usually come with? And essentially, what are the other organisms that might be related to or associated with or even co-regulating the biology of different microbes in different places in the world? And so that's one feature of these data. The other thing that this lets us do outside of planetary protection or outside of Mars is actually gives us a way to track the biodiversity of the world itself. And this is important because we know that, for example, in North America, 1 out of every 4 birds in the past 50 years has died, is gone. Some of it's because of habitat deconstruction. Some of it is just the stress in the environments, dwindling populations. But we know that this sequence information is not just a microbial map, it also picks up other organisms, including eukaryotes like birds, other insects, and it gives us a way to map the diversity, or in some cases, even the lack or missing diversity that can appear. And the reason this is important is not just because of mapping and collecting stamps, but it gives us a quantified metric of what's in the world around us. And we know that there are times when things change. So if you look at basically the history of the Earth in the past 500 million years, there have been multiple times where there have been probably 5 massive extinctions. And this has occurred in the world that has without polar ice caps. You can see when it was very warm. And in a world like we have now, where the world does have polar ice caps, you can see we're here right now in blue. And 5 times throughout Earth's history, we've had massive global extinctions. You can see here anywhere from 75% to as much as 96% of all species were lost. And species get lost whenever you see big changes in either down or up of the Earth's temperature. And so we know that this cataloging of the genetic diversity in the organisms on Earth is something that is very precious, because the history of life on Earth is pretty rough. 99.9% of every species that's ever lived has died. But there's a danger now, because they see it's starting to go up again at kind of a straight line. We don't want to create a 6th extinction, or at least I certainly would not like to have that occur. And what's interesting about this timeline, again, this is 500 million years, everything that's happened on Earth and the temperature and extinctions. But if you kind of take even a bigger step back and say, okay, let's take this 500 million years and let's just kind of zoom out even more and put this snapshot of time into the broader, complete timeline of the Earth itself. And so if you look at where we are today, we're in an Earth that has liquid water. We're basically in a very nice but very temporary temperature canyon. This is where we are today. It looks like we're going to have liquid water, but the Earth used to be much hotter. And someday when the sun engulfs the inner planets, it'll get even hotter again still. And so what's interesting about this is at some point, we'll get to maybe about 1 billion years, the sun will actually start to increase its luminescence. It will literally boil the oceans. And so, I used to think that we had 4.5 billion years until we lost the Earth, but we really only have probably a good 1 or 1.5 billion, which is still a really long time. But from my perspective, I kept thinking, well, I just thought we had so much more time. And so I think, looking back at the changes in the ecosystems and the organisms and this really genetic cataloging of the unique species, we used to always think about, or at least when I was a kid, I was trained that, there are 3 kinds of organisms, right? There's producers, consumers, and decomposers, and that, this is basically it. But I argue to you today that we represent a unique kind of species, a 4th and distinct type of organism, in that we're the only ones that are aware of this sort of food chain's frailty, this ability of it to be destroyed. We're basically the only ones that can guard against this sort of mass extinction. Of course, only humans can prevent extinction. Sometimes we have caused extinctions, which is part of the problem, but only humans are extinction aware. We're the only ones who can act on this knowledge, and the only ones that can actually prevent it from happening. And so I think in this regard, this gives us a very unique duty to the universe, I think quite literally, and also to the Earth and to all.
Chris, we can't hear you. Hello?
Oh, can you hear me?
Okay,
Can you
it's
see
better
me?
now. I can hear you now.
I was just at the very end punchline. That's just my connection's unstable. Okay. So I do make this case here in a book that just came out, is that I think this is actually preserving and mapping genetics for helping this planet and someday eventually others, is what I argue in my book. And so with that, I want to thank really everyone in the laboratory who's made this work possible. Deep gratitude to everyone in the Metasub global network, particularly Dr. Nagarajan has been a fabulous collaborator and colleague in this regard. And we've recently published multiple papers, some of which I'll highlight today. And of course, really wouldn't be possible without the work of all the members of the lab And the biggest thanks is really to the patients and their families who donated blood, sweat, tears, and even their bodies, and to really all they leave behind, because even the sewage left behind itself is informative and that we've been using in really extraordinary new ways. So with that, I am really happy to take any questions, and thank you so much for your time.
Thanks a lot, Chris. That was an amazing exposition of all that genomics can give us, and also essentially our responsibility to catalog the genomic diversity around us. We have a few questions, and I have a question as well, but let me start with the questions that are on here in the Q&A box. You can see that. So the first question is about SARS-CoV-2 in the 2019, I presume, meta samples. Like, take that.
Yeah, did we go back and see if they're there? So we have looked and we don't see them. So it's a good negative control. We recently also got some samples from cats. It's a project we have called Metacats, actually, which is actually, if you go to Metasub itself, the homepage, there is, I want to pull this up here. Let me share my screen. I can show you this. We have a range of different projects, and in particular, one of them is, you can see here the different projects. There's many of them now, including sewage. One of them is Metacats, is looking to see what's the differences in carrying capacity for different viruses in domestic and feral cats. And what's exciting is, this is a carrying stool and saliva, and we have some samples banked from 2019, and you can see here all of us working on the Metacats. And so collect samples and we analyze what's in cats, and they also have been negative. You can see here we made the stool sample look a little bit like ice cream because we thought that would be easier to handle. But yeah, so they were also negative from what we've seen so far.
Okay, so we have another. Thanks, Chris, for that. The next question is from Dr. Shyam Pravakar, a colleague of mine. It's about, is it possible to do experiments in space? Can humans or robots in spaceships culture cells, perform basic assays?
Great question. So actually, very much so. Yeah, there is. I can even show you, although since we've got a little bit of time here, I can show you an example of that where we've actually done and performed sequencing in zero gravity, which worked out quite well, actually. So I will do a quick demo of this. I didn't talk as much about the astronaut genetics, but we did have a project. Let's see, let me share my screen. With Kate Rubens, who went up there. She's a trained virologist and went up, and it would just be, of course, more convenient to sequence on the space station. So we launched a mission called the Biomolecular Sequencer, worked with Aaron Burton and others at NASA to say, well, let's get a patch and see if you can take a nanopore sequencer up into space. And the first place we tried it, though, was on the parabolic flight simulator. In this case, we tried it first with some pipettes. You can probably see Andy Feinberg there. Basically trying to do pipetting in zero gravity. And you can see the tips flying all over the place. You can see also the tubes not staying in place, and he's trying to maintain his composure. And while you're in zero gravity, getting it to work, you can see the tubes zooming around and eventually crashing back down. Kept his composure. But we did get it to work and got the nanopore working in 0 degree, and it kind of passed the first test in October. And it up there in 2016 and safely made it to the space station, and we showed that it was possible to sequence in space for the first time successfully. And so that was it was indeed possible. That was a great project with NASA as well. And there's now some cell culture up there, but it's a little bit harder, as you can see with the tips. So now we just do 3 tips at a time in a bag. We don't do tip boxes.
All right, great. That was a very interesting video. Okay, so we have another question from Sham about, are there any novel insights into human genetics or migration or behavior from the meta-sub data, given that, we have data over the years, and I know we have the Olympiome related projects, perhaps you have.
Yep, great question. So we've seen, some of the advantage of longitudinal data is that you can start to look for new trends. Some of the things we've observed during the pandemic is that the amount of human RNA that showed up in the subways decreased over time, and the diversity of species, the number of species and the diversity all decreased, particularly for things like Cutibacter acne, which is a very common human skin commensal. we could see less of it because there's less people. But we could see this enrichment for other more, stone dwelling or some of these core microbiome species that were not associated with humans. They started to increase a little bit. So we could see some of those changes. But for migration, we're just beginning to look at that because we had samples before and after the Olympics in 2016. We're collecting samples across Tokyo for the 2021 Olympics as well. And this will give us a really unique opportunity to characterize what happens, really around, as people migrate around the world. We've seen in Rio that the diversity increased, so the number of species and types did increase during the Rio Olympics. We don't think we'll see as much of that for Tokyo because of so much of disruption and limited travel, but we'll find out. And so, yeah, these types of data let us answer, kind of very unique questions in that way.
All right, so we have another question from my colleague, Dr. Swain Chen. The question is, can you talk about the current conversation about expectation of gamer diversity on Mars, especially in the context of ideas about panspermia? Might it be that gamers would be similar in profile? And do you have ideas on how to prove that if that's what the data comes out as?
That's a great question. So we, because there's always this question, if you ask a lot of exobiologists, they'll say, okay, do you think there's life beyond Earth? And most exobiologists or astrobiologists that I know, almost all say yes. They think there'll probably be some microbe out there somewhere because microbes can survive pretty much anywhere on Earth. And so will there be intelligent life is kind of an older question, like the Drake equation is a classic formulation of this challenge. We mostly don't know, but the thinking is that if we did find life and it was DNA based, that it would probably have some degree of a Kamer profile that's similar. For example, we wouldn't find a 95% GC composition organism, unlikely. And now we don't know that, but that's what I would speculate, is that because it is hard for an organism to survive on Earth that way, and we've not seen anything really beyond 70, 75, or even 80% GC content, I don't think we'd find something too far beyond that elsewhere. But now that being said, there are these interesting sort of non-MERS, the DNA sequences that never get used, basically what's called the distribution of overrepresented or underrepresented Kamers around the world. We know that they, there are, some that have just never been observed. And so maybe if we go to Mars, we suddenly find these Kamer sequences that have never been utilized on Earth, suddenly they're possibly, but I would guess not, even with or without panspermia. For those who don't know, panspermia is the hypothesis that life originated from one plus and spread elsewhere. Could also be through collisions of asteroids, collisions of protoplanets, and then essentially ejected matter from one planet, projected onto another and then landing there. So it doesn't have to be necessarily even by someone purposely doing panspermia. It can happen accidentally. So yeah, that's my thought so far. Interesting question.
Sorry. Another question from Anton at Emory. Do you think that the public transport could be an important source of airborne virus spreading? Can you compare its importance to offices or other public spaces?
So we do know this is still debated. This is really the key question that came up in the pandemic. And actually, Naran, I think you presented at one of the MetaSub meetings some of the early slides. You had a really great slide deck of some of the very early literature on this, a great summary of what was there. I think it was back in April of last year. I don't know if you remember this. You had a really great summary of what was the literature at the time. It's been hotly debated because it depends so much on the humidity and the overall airflow and the temperature of the room for all these places. But we know it has been transmitted, but we know that I think the fomite transmission risk is very low. I think the viruses, once they land, they degrade quickly. We've done infectivity studies for VRE6 cells. Nothing that we've ever tested from a surface, even in hospitals, has been able to infect a vero cell. Now it's an artificial cell, but still, I think that risk is low, and that's been validated by other papers. But the airborne risk, which is what you're asking about, it is high in all those spaces. When you have someone actively coughing as close to you, we know that it's happened on planes. It's happened in all those places. The biggest thing we've seen that just reduces that risk is just ventilation, just opening the window, or even if you're just staying outside versus being inside, it's just amazingly effective. And so, which one is the worst? Like you're kind of saying, is it better to be in a subway or a bus or an office? It really just depends so much on the airflow. And I think one of them, I wouldn't categorically say one is better or worse than the other, but really just anytime you're in a closed environment with limited airflow, it is a risky environment for airborne transmission.
All right, thanks. We have a question from an anonymous attendee about the likelihood of false positives getting picked up from the Mars sample return project. I misidentifying previously unknown human microbiome organisms from NASA geoengineers as Martian microbes instead.
It's about a 50% chance, I'd say, based on all of our prior work. So it is, but so that's why we have to be so careful, is that we'd want to look at the camera composition and say, well, does it look a lot like Earth? And maybe it is from Earth, or even though I just told you that it might be the same on Mars anyway. But that's why I'm hoping that by the time we get to 2032, we'll have a much more exhaustive profiling of the diversity of life on Earth. So I'm hoping it'll be, instead of 50%, maybe 10% chance. But today I would put that number at 45 to 50% chance of a false positive, I'd say. And I'm hoping that by 2032, it goes down to 10%, maybe 5%. But it's not going to be like 0.01%, because there's so much life that we still have not mapped or found on Earth as it is, I'd say.
Do people have any thoughts about, let's say we do find something that matches, you can find a homology, let's say, and how do you distinguish whether it was a contaminant or whether it's an example of panspermia? Is there any other ideas of how you could discover that that microbe has been growing in the Martian environment, for longer and has been evolving in the Martian environment? Would it be something to do with its transcriptomics? Would it be epigenetic markers? What else could we think about in terms of catching that?
I don't know if you're asking the general audience, but you touched on something I was about to ask, is that the base modifications I think would be distinct, because the types of radiation are present are higher or different, both in some cases. They would be repaired differently potentially in that environment. So I think if we looked at both genetic and epigenetic differences, I think that might be the best sort of suite of clues that would be able to discriminate between really terrestrial versus Martian origin, I think. But we'll have to see. I actually also look forward to the point where people are on Mars or the moon sequencing something as things evolve, then you send the data back to Earth, and we then synthesize that genome and then start to characterize something that's basically a novel microbe that came from another planet, but you don't have to transport it. You can just, send the sequence data, which would be cool.
Are there other thoughts about, if you did acquire, sorry, there's no more questions, I'm just going to add a few of my own at the same thread. If we did find microorganisms on Mars, and if we could culture them, are there thoughts about how to bring them into an Earth-like environment to see if they could survive?
Well, we've done some of this, and I didn't go into too much detail, but we have done this for something called the Extreme Microbiome Project, which is a kind of a related project to, or a lot of the same people to Metasub. Is there, for example, for the here, actually I'll pull this up here, is the Extreme Microbiome. We actually have this project where we've been characterizing organisms from really unique environments. One of them is Lake Hillier, which is this bright pink lake here you can see. And to get these, and this is also a sample taken from a canyon, you can kind of see the different places where we've been collecting samples. But if you go back to this one, we had to use some of the water, this pink water that had these Delina sort of Selena organisms, and spike that in to do the culturing. And only then would some of the organisms actually grow. So we may have to do the same thing for this soil from another planet, is to kind of sprinkle it in, because there may be small molecules, peptides, things we don't understand but are present, that are required for growth, and we've already seen that on Earth. So I wouldn't be surprised if we had to do the same thing for weird microbes from Mars.
So there's a few points. I actually didn't notice that there were some points on the chat.
Yeah.
If you want to see, is there a topology map of how genetic materials change with gravitational force in space? And I guess it really, yeah.
Not yet. Well, the gravitational fields are ironically very close to what they are on Earth in terms of the amount of gravity. Because the distance is still not that big, and when you don't ever have really zero gravity, you just are falling at the same speed as your spacecraft, so you still have gravitational fields, you just have weightlessness basically. So I think, but we haven't seen how it changes too much, nothing obvious. It's hard to disentangle it because we see so many new things just on Earth, so to say is it really new just because it's in space, we can't say yet. But so I don't know yet, or we don't have any evidence yet that there's a big difference, not yet. And then the other thing was protein structure. Now, that's a really good hypothesis. Would you get more, if you have either greater pressures? Well, on Mars, it's only 1% of our atmosphere, so it might be less, or there the protein structures might be able to have to accommodate less atmospheric pressure, lower temperatures. So protein structure would also likely change. It's a really good hypothesis, I think. Yeah.
All right, I think we are at the end of the questions. I'd like to add one last question. I was very intrigued by that correlation with antibiotic usage and the air microbiome, or AMR elements in the air microbiome. Why do you think that happens?
I think it is, and it's an association. It's I think it really is this shedding in the environment of when people are taking antibiotics. It's a selection pressure, and that is the response of the organisms. Basically, it's creating enrichment for that resistance gene. So it's really just like if you do enough antibiotics on a plate, eventually some of them become resistant, and the usage of them around the world are different enough that it leads to, I think, a differential selection pressure.
All right. Thanks a lot, Chris. That was a great talk and a nice set of questions and answers. It was a good discussion. I hope we can have you again in person, hopefully in Singapore in the near future. And with that, thank you. Thanks again, and see you.
Thank you very much, and Dr. Nagaran, fabulous to be here. Thank you so much, and thanks for being a great collaborator for all these projects. Thank you, everyone.
Thank you. Take care, guys.
Thanks.